Professor SNAB, where we aim to make A-level biology as simple as a cell. So today we're looking at another core practical which investigates the effects of temperature on the development of organisms such as seeding growth rate, brine shrimp hatch rates. So the following practical will be looking at the effects of temperature on the development of simple organisms such as brine shrimps. So what do you need for this experiment? You need brine shrimp egg cysts. You also need sea salt, dechlorinated water, salt water, beakers, water bath, stirring rod, magnifying glass, forceps, pipette, bright light, refrigerator, and graph paper. So place 2 grams of sea salt into a beaker containing 100 centimeter of dechlorinated water and dissolve in the water. Put some eggs on a sheet of paper so that it's easier to count, and wet a piece of graph paper in salt water and place it on the paper to pick some eggs. Make sure you count the number of eggs you've picked up. And place the graph paper into a beaker and wait for the eggs to detach into the water. Incubate the beaker at a set temperature between the temperature range of 5 to 35 degrees, and using a bright light, remove the hatched larvae using a pipette and count. Repeat this for the remaining temperatures. So what is the risk assessment of this practical? So there's the risk of broken glass. So there's a possibility of cuts from sharp objects. Keeping glassware away from edges is really recommended. And in case of an accident, elevate the cuts and apply pressure. Do not remove the glass from wound and seek further medical assistance if needed. There is also the risk of hot liquids because you are dealing with a range of temperatures. So there's the possibility of burns. So risk can be reduced by wearing eye protection and using tongs to remove boiling tubes. In case of burns, run under cold water and seek further assistance. And finally, you have biohazard, which is the possibility of contamination from the shrimps. So use disinfectant before and after the practical. So the results you obtain, plot them on the graph with temperature on the x-axis and the number of hatched larvae on the y-axis. So looking at the conclusion, so an increase in temperature leads to an increase in the number of shrimps hatched up to the optimum temperature, after which the number of shrimps hatched decreases. Hence, we can conclude that with an increasing rate of climate change, the development of brine shrimps will decrease. So you can modify the practical to look at the effect on plant species, and this can be done by looking at the number of seedlings that germinate at different temperatures. But you will see a similar trend where the number of seedlings germinating increase with temperature up to the optimum temperature, after which it decreases. And this suggests that plants are also affected by climate change. So that is all from Professor SNAB today. Hope you found this topic useful. And if you have any questions, let me know in the comments below and I will get back to them as soon as possible. And don't forget to like, share, and subscribe.